begin recording. Now, if I go and I look at when we're supposed to have our first test, it should be, I believe, next week, because 8 through 12 is spring break. I'm going to double check with the other professor, but I think I'm going to probably open it up over like 5th, 6th, 7th, something like that. Now, if somebody has like some sort of reason they can't do it on those days, that you need to, like, if you know that you're going out of town, something like that, you can let me know if I need to open it up for you early or something of that nature, but it's going to be completely online. When I was looking earlier at a review from exam 1 from previous years, it doesn't even show topic 5 in here. So I'm going to confirm, but if this is the case, then it's only going to be topics 1 through 4, which means it is DNA structure. Topic 1 was DNA structure. Topic 2 is DNA replication. Topic 3 is transcription, and topic 4 is translation. Next week, we will start talking about Mendelian inheritance and that sort of stuff. So that's chapter 3 and 4. But the majority of chapter 4 is summed up in this video. It's a TED Talk about how different creatures do sex determination. And then I have you start working with some virtual labs that are going to get you ready, because we have fruit flies that have arrived while we weren't at the school, and they have red and white eye colors that are X-linked, and we are actually going to then pull the DNA out and PCR it and run it on gels to be able to see what type of mutations have happened to cause these eye color differences. So all of that stuff going on next week is to get you ready so that when you come back from spring break, we can jump right on in and do lab stuff. But I'm going to double check with the other professor, but it looks like chapter 3 and 4 would not be on this test. So that would still give you a whole week to study before you got there. So again, I will post those results from talking with the other teacher, just because I want to make sure that that seems fair. So the review kind of things you need to be familiar with. Kind of like if you went through tonight's topics and you felt okay, then you're probably in pretty good shape. But if you're going, oh, I don't remember about transformation, or you didn't learn it, that kind of thing, then you need to go back and work on those stuff. So topic 1 is bacterial transformation, like plasmids, antibiotic usage, like why we put in the antibiotics, that kind of stuff. Number 2 is about plasmid maps and their features. So again, like those worksheets, if I give you a plasmid map and I ask you, okay, you're going to digest with these enzymes, what size fragments are going to be produced? Step 3 is for topic 3, DNA isolation. So major steps, chemicals. That's more for kind of knowing how we did it. I'm not going to ask like specific chemicals or anything on that, but just the remembering how cells work, and that you would need to split apart to like get the chromosomes away from the plasmids and that sort of thing. Part 4, DNA, RNA, protein structures to the level of strand direction, but not expected to draw them out in detail. So if I gave you any kind of pictures and had you label them, they're going to be pictures on the level of like this, and like before, where, let's see, like this. And if I'm like, okay, what is this thing here? Like it'll be very simplified pictures. They are not going to be very complex. I guess maybe the next picture would have been better. Probably one closer to like that, that you could, if I gave you a replication fork and said to label out parts of it, that you could do that. kind of thing. But I'm not going to expect you to draw out like how many rings are in guanine or something like that. Like, no. So gene organization, talking about like where the promoter is, where the coding region is, but don't memorize like specific sequences. Like the consensus sequence is TATAAT. Yeah, like no. But just promoter, coding regions, no, like, because it's different for transcriptional start site versus translational start site. And I'm going to ask you some questions. It's like a true false question. And if it's false, you need to correct the wording to make it true. So if I say transcription starts at the start codon, you need to be able to cross out transcription, say translation starts at the stop codon or at the start codon, something like that. Or change out start codon to say the +1 site, the steps, enzymes, proteins involved in DNA replication, transcription, and translation. So the main players, like we say, helicase, large, small subunits, that level of understanding. You don't need to know the specific subunits within the subunits or specific elongation factors, anything like that. The most specific I will ever ask is like DNA or polymerase 3 versus 1. Because we talked about the different jobs that those did. So going back to replication, how polymerase 3 is the main workhorse, and polymerase 1 just comes along to remove the little RNA primers and replace it with DNA. But everything else is done by DNA polymerase 3. So we don't even talk about 2. We don't even talk about the eukaryotic versions, anything like that. Be able to write out DNA sequences to translate it into mRNA and then go DNA to RNA to amino acids. Don't memorize the codon chart. I will give it to you. But just if I give you a strand of DNA, can you tell me what the matching strand of DNA is going to be? Can you then turn it into mRNA? So make sure that you understand how the directions work on 5' to 3', that kind of thing. Number 8 is restriction enzymes. So their function, where they came from originally, that kind of thing. Number 9 is about gel electrophoresis and like being able to read a gel. So you have some virtual labs on that this week. Just that I'm going to give you a plasmid map and say, if you digested it with these enzymes, what size fragments would you get? Okay, now draw your answer on this gel, and you need to be able to put lines at the appropriate size. Understanding about consensus sequences and SNPs. That's going back to like the dog hair paper. And that the closer something matches to a consensus sequence, the more likely it is to be read or turned on if you're talking about like promoters. And SNPs, how they are just a single nucleotide change, as opposed to like large scale deletions or anything like that. You need to know the meaning of chi-square analysis and p-values, but I'm not going to ask you to do a chi-square analysis. Like you're not going to do the calculations, but if I say, what is the cutoff for p-value, you should be able to answer that. Or if I said they got a p-value of 0.05, is this significant, yes or no? I'm going to, I think, talk about model organisms, or I'm going to move that to the next test. Usually we would have been doing the model organisms during class because I want you to get to play with the flies and the nematodes and stuff. But we did talk about some model organisms today with the transgenic animals. It's basically that you want to use animals because they can reproduce faster. You'd want to pick an animal like that you could house cheaply, or you could put a bunch of them together in a small space in the lab. That's going to be a lot better than trying to do elephants or something like that. You'd want to pick something that's got some kind of variation on it, because obviously if every single creature is an exact clone, then that's going to be very hard to study how a gene changes things. But there's a lot, just kind of thinking about the benefits of using model organisms. And then going back to our case studies. So if you remember back with the bacteriophage viruses, where we were tagging phosphorus and sulfur, and kind of the main takeaways from those kinds of things. So yeah, it's all fair game for the test, but the case studies are meant to help you understand the topics that were talked about in the book in more detail. Like how today's case study talked about FOXP2, my questions aren't going to be about FOXP2, but because we reviewed on plasmids, we've done review on transcription and translation and all of those kinds of things. So, are there any questions? Oops, I didn't realize there's a chat there. Oh, the test is going to be some short answers, some multiple choice, some fill in the blank. Usually we did it to where most of the test was online, and then you had a few questions that you did in person. I'm not sure how we're going to do that since we can't appear in person. We might change some of it. It was usually that we would do like the questions that had like figures, like draw this out. We did that in person so that you weren't trying to like draw in Microsoft Word, that kind of thing. Anything that's easier to do like by hand, we would have you do that in person, but it would be like 80% of the test was online and then 20% of the test was in person. Since we can't come in person, I don't know, maybe that'll just get rolled on to, I hate to dump it all the way on after spring break, because by then you're like, wait, what topic? Like, I don't remember how to do that. So I'd rather it not have that big of a gap. It might just get moved on to test 2 or something. We'll have to figure out how to do that. Again, I'm going to talk with the other professor to see how she wants to do it. But yeah, the questions will be kind of closer to like the post lab or post lecture quizzes. Like I have to release this week's quiz. So yeah, it's just kind of assuming that you've worked with it, you know the vocabulary words, that kind of thing. But yeah, combination, some multiple choice, some short answer. And by short answer, I mean like might only require a couple words to answer it, at most needing a couple sentences. Sometimes it's like list 3 ways of something, so you don't even need to answer in full sentences. Or it might be like use these pictures and put them in order, kind of things. So yeah, things that most of it you can answer without even needing to use complete sentences. As we go on to further topics, some of the other tests will have more like, okay, it might take 2 or 3 sentences to answer it. But again, I'm assuming that you're kind of running on a time limit, and if you can answer it with bullet points, then answer it with bullet points. I don't want you freaking out because you wrote an essay when you needed 1 sentence. Like, go simple. Trying to think if there's anything else. Yeah. Any other questions? You can always email me. I'm gonna figure out a way on how we're gonna submit the other stuff. What I'm gonna take as a grade for you. Let's see here.